Hello everyone, my name is Alex Wu, and today me and my cohort shall be presenting the findings discovered in the analysis of Case 2. I'm also joined by my fellow peer Tasmia Islam and Llewellyn Wise. Case 2 represents the administration of a 14-year-old girl into the emergency department after she was found to be suffering from onset of symptoms including neck stiffness, rising body temperature, and an emerging rash. A spinal tap and blood extraction was produced and transported to the laboratory in order to identify any pathogen that may be located in her cerebral spinal fluids and/or within her bloodstream. A throat swab was also conducted to identify any suspicious flora located within her mouth. Laboratory finding describes the CSF as slightly turbid, with large traces of white blood cells at around 280 × 10 to the power 6 cells per liter. A photogram stain identified only the presence of pus cells within the CSF, indicating the presence of an inflammatory response but without any bacterial presence. Blood culture conducted on the collected sample was found to be infected with colonies of Neisseria meningitis. This was followed by most probable number tests which showed that only half of the glasses were turbid, indicating that N. meningitis was quite significant. The throat swab result found traces of different flora with Streptococcus viridans being among them. However, we do not believe it to be the causative pathogen, as it is a normal flora found within the mouth and not detected anywhere else. As such, this bacteria was deemed as a contaminant. Neisseria meningitis, also known as meningococcal, is a gram-negative diplococci that is known for causing meningitis. And meningitis can be naturally found in the nasopharynx cavity. However, an increase in its concentration can allow the bacteria to penetrate into the bloodstream, from which it begins to multiply and travel to the spinal cord to infect the meninges. And meningitis is highly contagious, and especially to children between the ages of 2 to 18. The disease utilizes two forms of virulence factor, from which it creates either a biofilm or capsule. The capsulated Neisseria species contains the antigen mostly utilized by the vaccines, but Group B capsules are wholly immunogenic, as their antigen markers closely resemble those found on normal body cells. Meningitis is a disease characterized with the inflammation of the meninges, a protective membrane surrounding the brain and spinal cord. Meningitis causes an array of different symptoms, including stiff neck, headaches, nausea, fatigue, and if untreated, will lead to seizures and death. Our group strongly believes that N. meningitis was a causative agent for the young girl's illness. The symptoms presented by the girl matches those that are closely associated with the disease. Also, the presence of N. meningitis found within her bloodstream indicate it has broken through the nasopharynx's walls and become very pathogenic in a septic state. Here's a flowchart dictating a list of microbiological tests that were done to verify the hypothesis. Day 0 had already been performed and documented in the previous slides. Day 1 begins with utilizing the colonies from the blood samples for a gram stain and oxidase catalase test. If the results correlate with characteristics of a Neisseria species, then a follow-up inoculation will be done onto a New York City medium agar and chocolate blood agar for differentiation. Further inoculation will have been performed on a molar Hilton agar for AST testing using custom antibiotic layout and a purity plate for API NH. This section of the slide will describe the methods used in more detail to present the reason why they were undertaken. Firstly, an initial analysis of the CSF serum was conducted to note down various factors that may be found within the fluid. This can include glucose, protein, red blood cells, and white blood cell concentration. The identification of these factors would indicate if the spinal cord is infected, as none of these factors should be found in the fluid at all. Macroscopic analysis also provides a clear indication of the CSF state, as turbidity would indicate an infection.
To grow the suspect pathogen, we will plate it out on a range of plates used to identify Neisseria meningitis. These plates are a 5% CO2 blood agar, chocolate agar, and New York City agar. The colonies will be plated out under aseptic conditions, and the plate will be placed upside down as to ensure any condensation on the lid does not drop and move the colonies around, being sure to label the plate, not the lid, as the lid may be misplaced. A gram stain will also be performed with pathogen isolates. Colony will be smeared onto a plate, heat fixed, then treated with crystal violet for 1 to 2 minutes, iodine for 1 minute after that, acetone for 5 seconds, and then a counter stain with carbol fusion. Catalase is an enzyme which produces oxygen and water from hydrogen peroxide. First, we will do this procedure on positive and negative controls to check whether the reagent is working properly or not. Then, take a stick and take some colony from New York City or chocolate, as the unlysed red blood cells may produce a false positive. Next, put it in a test tube and add a few drops of catalase reagent to it. A positive result will produce bubbles and a negative result will not produce bubbles. To determine the presence of cytochrome C, we will perform an oxidase test. First, we will perform this test on positive and negative controls. Then, take a small piece of paper and place it on a glass slide. Next, collect a few colonies and smear it on the paper. Add oxidase reagent to it, then wait for 15 seconds for color development. A positive result will turn blue or purple and a negative result will have no color change. A number of organisms have specific antigens which can be tested for with an agglutination test. Some of these organisms include Streptococcus pneumoniae, Neisseria meningitidis, Haemophilus influenzae type B, and Streptococcus agalactiae. When a sample containing certain microorganisms are mixed with their reagent, then specific antigens present on the microorganism surface bind with related specific antibodies. A clump is created which can be seen with the naked eye. To perform an agglutination test, we will heat the sample in a water bath for 3 to 5 minutes at 80 degrees Celsius. Cool it, then centrifuge for 10 minutes so we can dilute the sample to prevent too much clotting. After the dilution, put 1 drop of each reagent on a separate ring on the test slide. Then, put 1 drop of preheated sample onto each reagent on the slide and mix it thoroughly and gently with a disposable loop, making sure to do a positive control. Use a new loop for each drop. Roll the slide and read the result. A positive result will yield clumping and a negative result will yield no clumping. We will also perform an API-NH test. This test specializes in the detection of Neisseria, Haemophilus, and Moraxella species. The test itself is relatively short and requires about 2 hours for the results to be observed. An added addition to this test is that it also utilizes far less reagents than an API-20E. To determine the best course of treatment, we will conduct a series of antibiotic tests. This will allow us to provide the best antibiotics that should be used to treat the pathogen and show any possible resistance the organism might have developed.
Expected result of Neisseria meningitidis. Cytological test of CSF. Bacteria shows a typical abnormalities feature which are associated with bacterial meningitis. It is starved with high cranial pressure, WBC count is more than 10 cells per millimeter cubic, low glucose concentrations and high protein concentrations. Blood agar 5% carbon dioxide. It shows round, smooth, moist, glistening, convex with a clearly defined edge, gray color and non-pigmented. Chocolate agar 5% carbon dioxide. It shows circular gray, more than 1 mm, creamy colonies. In New York City agar, it shows circular gray, raised and 1 to 2 mm colonies. In gram stain, it is gram-negative diplococci, intracellular, catalase test positive, oxidase test positive, agglutination test, agglutination formed, and API-NH, it shows result for Neisseria meningitidis. In antibiotic sensitivity test, it is sensitive to ampicillin or penicillin, ceftriaxone, chloramphenicol, rifampicin, ciprofloxacin, and meropenem. Risk factor of Neisseria meningitidis. Neisseria meningitidis is known to target teenagers and young adults. It has lack of vaccine due to being immunocompromised or insufficient vaccine doses, drastically increased infection rate. It has low memory cell count from previous vaccine. It is risky in school environment, animal contact, location of recent outbreak or infected individuals. And also the vaccine is available to only some strain of bacteria. Most non-encapsulated strain are undetectable. After finished all the tests, next step is storage of sample. It is often necessary to re-examine specimens. In order to do so, specimen need to be stored. Depending on how long the organism needs to be stored, there are a range of long and short-term options. Short-term storage is when the organism needs to be stored for several days up to a few weeks. Method used: Dorsan transport media up to 3 weeks, chocolate agar slants up to 1 week, silica gel package at 4 degrees Celsius up to 2 weeks. Long-term storage is when the organism needs to be stored for a longer time than a few weeks. Method used: freezing, lipohylizations. Bacteria can be stored at 4 or -20 degrees Celsius, can be done without refrigerations, but it is expensive. For a laboratory technician, one of the primary responsibilities is to maintain a regular quality control checklist, as any clinical experiment requires a certain degree of quality assurance. For meningitis laboratory, we need a designated personnel for monitoring the quality policies and ensure the implementation. Trained and motivated staff, equipment which is used in a laboratory, make sure it is works properly and gets proper maintenance, and it is treated and used properly. Purchasing good quality reagents and other material, everything should be documented and recorded. Provide a good customer service, and also laboratory should be aware and considerate of biosafety, basic safe operating procedure, and waste management. Laboratory errors often bring detrimental consequences for both patients and practitioners, as incorrect identification of pathogen results may harm a patient's health, and associated practitioners suffer legal and monetary consequences. At the patient level, it includes not getting proper treatment. Patient may receive unnecessary treatment, which includes additional expenses or treatment complication and a delay to receiving the proper treatment. Wrong choice of antibiotics and improper use vaccinations. At national level, it may cause a delay in understanding whether it is an epidemic or not, improper national control measure recommendation, or a delay in implementing public health measures.